to get here finally. Thank you very much, Anna, for this nice introduction. We've been collaborating, I think, more than 2 years, maybe 3 or even 4.
Probably not 4.
No, maybe 3. And we talk on Zoom regularly. So it feels like I've always been around, but I've only met Anna once before in person, and some of my collaborators I've never met. So really happy to be here. When I accepted the invitation, I didn't realize how difficult it is to get to Kingston. I guess the bird flies, the distance is not very long from Nebraska to Kingston, but the public transportation, it took the whole day to get here, but it was so much worth it. The day went by very fast, and I learned interesting things, and happy to tell you a little bit about what we are doing with DNA polymerases in cancers. And I will start by okay. It is somewhat frozen.
Start over again. Try now.
Yes.
Yep, there you go.
Okay, so I will start by explaining why I'm so fascinated by this issue of DNA polymerases. This is a picture of Victoria, my former technician. This doesn't. Yeah, here we go. And her twin sister, Valerie. So these two girls used to be a single cell. And once this cell divided, one half went through this astronomical number of cell divisions to produce Victoria, and the other half went through the same astronomical number of cell divisions to produce Valerie. And you can see that there are very few errors during these cell divisions because the girls are virtually identical. So this is an amazing accuracy. And for my entire career, I've been exploring how this accuracy is maintained and what happens if something goes wrong and there are replication errors that lead to diseases or other things. So this is the pathway that maintains the accuracy of synthesis in all organisms. Most of the fidelity lies with the DNA polymerase. Polymerases usually selectively incorporate correct nucleotides. They very rarely make errors, approximately once per 10,000 or 100,000 nucleotides. And if this happens, replicative DNA polymerases also has an associated exonuclease activity that can excise the incorrectly incorporated nucleotide from the primary terminus and give the polymerase another chance to incorporate the right one, which it will most often do. And this would improve the accuracy of synthesis by about 2 orders of magnitude. And I have to say that the actual error rate can vary tremendously depending on the type of error. I just wanted to give you some numbers to stare at. This is maybe an average error rate, but it can vary by several orders of magnitude. And then if exonucleolytic proofreading fails, then there is a mismatch repair system that will scan the newly synthesized DNA for mismatches now in double-stranded DNA, and it will excise the portion that contains the error and replace it with a new DNA, improving the accuracy by another several orders of magnitude. So we end up with this number. Our genome is approximately 10 to the 9th nucleotides. So we get one error during every cell division, but most of our DNA is non-coding DNA. So this is the accuracy that we can live with. When things go wrong, there is no mismatch repair. Mutations in mismatch repair genes lead to hereditary predisposition to colorectal cancer and some other cancers in a syndrome called Lynch syndrome. This is also a syndrome that displays microsatellite instability, or MSI. You might have heard this term because mutations in microsatellite sequences are the most efficiently prevented by mismatch repair. So when it's not working, you get microsatellite instability. So this year actually marks, I think, 30th anniversary of the discovery of mismatch repair defects in Lynch syndrome. So this is an old story. My lab has been focusing for many years on this part of the pathway, where DNA polymerases must avoid errors, and if they don't, then bad things happen. So let me start by introducing the eukaryotic DNA polymerases. There are three that replicate the chromosomal DNA. And according to the most supporting model, DNA polymerase epsilon synthesizes the leading DNA strand, and DNA polymerase alpha synthesizes short RNA DNA primers at the beginning of Okazaki fragments on the lagging strand, and then DNA polymerase delta synthesizes most of Okazaki fragments. So basically, because the contribution of pol alpha is relatively small, it is a two polymerase, two strands model, pol epsilon on the leading strand, pol delta on the lagging strand. So the story that I will be telling today begins about 10 years ago. This is the first publication of the TCGA group on molecular features of colorectal cancers. And what you see here are mutation load in 224 colorectal tumors. Every dot is a tumor, and they are arranged in the order of decreasing mutation load. And you can see this abrupt jump between so-called hypermutated tumors and non-hypermutated tumors. This is a logarithmic scale, so this is a big difference here, and the majority of hypermutated tumors have mismatch repair defects. This was no news because we knew there were mismatch repair defects in human cancers, particularly in colorectal cancers. And also, I forgot to mention that many sporadic tumors will also display microsatellite instability, MSI, because of epigenetic activation of the mismatch repair gene, MLH1. What was new in this study is that the most hypermutated tumors here don't have mismatch repair defects. They carry mutations in the pol-E gene that encodes DNA polymerase epsilon. And the number of mutations in these tumors is really extraordinary. 500 mutations per megabase. This is approximately 1 mutation per 2 kb. So literally every gene is mutated in these tumors. And my lab has been most interested in how this amazing infidelity happens, what these mutations do to the polymerase to make it so inaccurate. Another interesting thing about this, so what I showed here were colorectal tumors, and then the same situation is seen in endometrial tumors. There is a larger proportion of tumors with mismatch repair defects, and they are hypermutated, and then pol-E tumors are the most hypermutated, and there is a term in the literature, ultramutated, to describe pol-E tumors, to distinguish them from mismatch repair deficient tumors that are less hypermutated. I don't know if ultra necessarily means more than hyper, but this is what we now use. It is a good way to have different terms because pol-E tumors are unique in many ways. There are also families that carry germline mutations in the pol-E and also pol-D1 gene that encodes the DNA polymerase delta, the lagging strand polymerase. And these people develop early onset colorectal cancers and sometimes other cancers and polyposis in their 20s and 30s. I don't know if you can read the text, but for example, for the family with this particular pol-E mutation, polyposis in their 20s and 30s, and colorectal cancer by 40 years of age with 100% probability. So this is a pretty severe syndrome. And the other interesting thing about these tumors, they have a drastically different prognosis and responses to therapies. Intuitively, you would think that tumors with so many mutations will be very aggressive, but this is not the case. They're actually very well recognized by our immune system because the large number of mutations creates a large number of neoantigens for the immune system recognition. So these tumors have excellent prognosis, especially in the case of endometrial cancer, to some extent in other types of cancers too. If they are excised, they practically never recur. If they recur, they respond really well to therapy with immune checkpoint inhibitors for the same reasons. They are very immunogenic. So there is an important task to classify tumors, and in many clinical facilities, there is now a tendency to not overtreat these tumors because these patients because these tumors would have excellent prognosis without any treatment. They may not need chemotherapy or radiation, and very often these are younger women who get POLE mutant tumors, so they would benefit from de-escalating the treatment. So it is important to classify the tumors as POLE mutant and monitor these patients, manage these patients differently. It is also important for people who carry germline mutations to know whether this is a driver mutation that is going to give them cancer later in life. So a lot of the work that we do is how we say disambiguation of POLE, variants of unknown significance. These hypermutated tumors have many mutations, as I showed you. So many mutations in POLE will also occur randomly and would have nothing to do with the origin of this hypermutability. So when you simply see a POLE mutation in a tumor, it doesn't necessarily tell you whether this is a significant mutation. POLE is an essential gene, so you can have a loss of function mutation. What you see is always a missense mutation, so you don't know whether it has any effect. And there are several ways to establish their functional significance. You can purify these proteins, study them in vitro, look at their enzymatic activities. You can model these mutations in yeast, which is a simple eukaryotic organism with DNA polymerases, very similar to human polymerases. There is a high degree of amino acid sequence identity, especially in the areas that comprise the active site, the polymerase active site, the exonuclease active site. So you can take mutations found in patients and make exactly the same mutations in yeast, and then ask whether they actually increase the mutation rate. It means whether they will drive hypermutability. You can do the same thing in human cell lines, introducing these mutations found in patients. And finally, you can make mice with these mutations and see if they develop tumors earlier than normally. Our lab does all of that, but by far the easiest and fastest way to say whether the mutation is functionally significant is to make it in yeast. And the advantage is that you are looking at the effects on mutagenesis in vivo setting under normal culture conditions. It is more biologically significant experiment than when you look at the DNA polymerase in a test tube. That I already said. So this is how it works. Again, you take a mutation found in patients, you make an analogous mutation in the yeast gene, which is called POL2, and then you take these mutant yeast, patch them on a plate, for example, that contains an antibiotic cannabinoid, and normal strains will not grow on this medium, but from time to time you will see colonies of resistant mutants. And this patch here shows you the normal wild type level of mutagenesis, and then if a mutation increases the mutation rate, it will increase the rate of cannabinoid resistant colonies. This is one of the tests that we do. We have more quantitative tests where we can measure the mutation rate precisely. You can also do this with any other reporter gene, or you can just simply sequence the whole genome of this yeast, which is not very expensive nowadays, and see if there is an increased mutation load. So by using these assays, we've now analyzed more than 30 different pol-E variants found in patients, either in sporadic tumors or in the germline of people with a seemingly increased susceptibility to cancer. And what we are looking for is whether these mutations increase the mutation rate over the wild type background here. And you can see a group that we call mutators. It means they increase the mutation rate, and non-mutators. And in this way, we can communicate to the clinicians that this mutation has phenotypic consequences. Now, I'm hoping to spend most of my time focusing on the mechanisms of these mutator effects. So before we start this, let's recall again what the main function of the polymerase is. The main function is to synthesize DNA. And this is done through the polymerase activity. The polymerase domain is responsible for synthesis, and then the exonuclease domain is responsible for correction of errors. So all the time, as the polymerase synthesizes DNA is being partitioned between the polymerase and exonuclease active site. When there are no obstacles, it refers to polymerase. When polymerization is slow, for example, the polymerase made a mistake, then it will transfer the DNA into the exonuclease site. And there must be, well, that's a picture for structurally inclined people. Here is showing how the two activities are coordinated. There's about 40 angstrom difference, distance between the exonuclease and polymerase active site. So this is quite a movement. Several base pairs need to melt to transfer the primer terminus from the polymerase site to the exonuclease site. And these two activities must remain in a certain balance. Of course, the polymerase activity must prevail, otherwise you will never synthesize anything. But there must be some amount of exonuclease activity for error correction, otherwise synthesis will be inaccurate. And this concept of balance was developed by an outstanding, and I'm pleased to say, Canadian scientist, Linda Rieger-Krantz. She spent most of her career at the University of Alberta, studying different polymerase mutants, and these are her diagrams that we like to use. There are some variants of polymerases, mutant polymerases, that have reduced ability to proofread, reduced exonuclease, and this will shift the balance toward polymerase activity and result in a mutator phenotypes. It synthesizes more, it proofreads less. Or you can have mutations that prevent efficient synthesis, then this polymerase will synthesize less and proofread more. It will be an anti-mutator, so it will reduce the mutation rate. But it will not make a good polymerase. Cancers would not like this polymerase because they need to synthesize DNA well. Now, what do the actual pol-E mutations found in tumors do? If you look where they are found throughout the gene, but I told you that most of them are passenger mutations, just from a high mutation rate. But there is very obvious concentration in the exonuclease domain. There are some recurrent hotspots, this P286R and B411L are the two most prominent hotspots. There are other recurrent mutations. Because they are all in the exonuclease domain, many people immediately thought, and myself included, that these mutations disrupt the exonuclease activity, and this is how they produce a mutator polymerase. And the flurry of papers appeared claiming that there are proofreading defects in human cancers, and even this syndrome of hereditary predisposition received a newly coined name, polymerase proofreading associated polyposis. And this was done before any biochemical activities were tested for these variants. And well, I thought so too at first when I saw how consistently these mutations hit conserved residues in the exonuclease domain, but something eventually did not quite match up. There are mutations at catalytic residues in the exonuclease domain that will definitely inactivate proofreading, and we never or very rarely see them in cancers. There are many mutations that would inactivate proofreading. We see none of them. What we see are some other recurrent hotspots, not anything that was studied previously in model systems. So we started with modeling the most recurrent mutation, P286R in yeast, which is P301R in the yeast gene. And this discovery was made by my very hardworking postdoc, Dan Kane, who is well, postdoc at the time, almost 10 years ago. He's now an associate professor at the university in Syracuse, New York, actually geographically very close location to where you are. So the discovery was that if you simply inactivate the proofreading activity of DNA polymerase epsilon, you only elevate the mutation rate threefold. But if you model this cancer mutation, this is a 150-fold increase in mutagenesis. So these mutations were doing something very different. So next we turn to biochemistry. How can I remove this? All right. I hope it doesn't disrupt.
I'll just no, maybe not.
That's okay. It's a small corner. It probably won't matter. Okay, so we turn to biochemistry, and this was done by my other very talented. We went too far. Yeah, so it's like that. Seems to be freezing sometimes. Here we go.
Yeah.
Started moving. Started moving. All right, here we go. All right, by my other talented postdoc, Sean Chin, who purified these polymerases and analyzed their biochemical activity. So now I'm going to torture you with a couple of biochemical slides. How this activity is analyzed. You take a DNA substrate, which consists of an oligonucleotide template and a shorter oligonucleotide primer. And the primer is fluorescently labeled. You incubate this with the polymerase. You don't give any nucleotides, so it can synthesize anything. It can only degrade the DNA. And when it degrades, it produces shorter fragments. And when you separate the reaction products in a polyacrylamide gel, your undigested primer runs here. This is a 50 nucleotide piece. And then any shorter fragments will migrate faster. So you see that the wild type polymerase produces shorter fragments. It means it can hydrolyze DNA through its exonuclease activity. The exonuclease deficient variant does not have any exonuclease as expected. And then the cancer variant actually had some, very much reduced, but had some exonuclease activity. And we also had a system where we can measure the accuracy of DNA synthesis by this polymerase in vitro. And the cancer variant was more accurate than the exonuclease deficient. So this is the situation opposite to what we observed in vivo, where this polymerase is very mild mutator, and this is a very strong mutator. So the exonuclease defect probably had nothing to do with it, and the polymerase was doing something else to produce a mutator phenotype. So that something else was an increased DNA polymerase activity. When we now incubate this substrate with the polymerase in the presence of nucleotides, it will synthesize to the end of the template. And then you again run a gel. Your unextended primer runs here at 50 nucleotides. And we are looking at how efficiently the polymerase will form longer fragments. And you see that the cancer variant is more efficient than the wild type and exonuclease deficient polymerase because it utilizes the primer faster, it disappears faster, and then it produces the full-length product also faster, and this is the quantification here. So it was a hyperactive polymerase. We also can do this test with a longer natural DNA template. This is a 9 kb plasmid, and we incubated with the polymerase in the presence of all polymerase accessory factors. And the synthesis goes around the circle, and we can quantify this again because it's fluorescently labeled, and the cancer variant is more efficient than the wild type or exonuclease deficient. So we thought that the pathogenicity of these mutations must be somehow related to this very efficient synthesis. And then we got some insight into the mechanisms of this increased activity by crystallizing this most common PR variant. And we collaborated with Erik Johansson at Umeå University in Sweden. And Erik is a world-class biochemist and a polyepsilon expert, and more recently with an interest in crystallography. But he also has one other very peculiar interest in building canoes. And unfortunately, I don't have a picture, like a really good picture that shows how beautiful the canoe is that he built himself from scratch from a piece of wood. But it shows you how functional it is riding in the rapids. Going back to what Erik did with the crystal structure, this is a part of the exonuclease active site of this proline to arginine mutant. And the DNA, when it is in the exonuclease active site, is supposed to be located here. But you see, when there is this arginine side chain here, it dwells into the same space that needs to be occupied by the DNA. So the arginine residue and the DNA cannot be there at the same time. So there is a physical block to the movement of the DNA to the exonuclease site. So the model that we came up with, so the wild type polymerase undergoes partition of the DNA between the exonuclease and polymerase site. This is, I told you, is quite a movement. So this is a slow process. So it synthesizes not very fast, but very accurately. With the cancer variant, this structure becomes impossible because the DNA cannot enter the exonuclease site. So the polymerase has no choice but to remain in the polymerization mode. So it goes fast. Even though when there is a mismatch that it needs to correct, it still will remain in the polymerization mode. And this is how you get so many errors. So this was with the most common variant. We have since looked at many others, and including many studied biochemically too, and my former graduate student just recently graduated last year did most of this work, and this just shows you the bulk of the work that she did, and I will spare you any more biochemical data, and I will just give you a summary of what we observed. So all these recurrent pathogenic variants were hyperactive polymerases with somewhat decreased exonuclease activity, although this was not very essential. Some of them had very minor defect in exonuclease activity, but they all had greatly elevated DNA polymerase activity. So what you end up with is not as much inaccurate polymerase as it is a very fit polymerase that is most optimally fit for efficient DNA replication. So this is what cancers prefer to have. And then you get the mutated phenotype and alpha mutation in tumors. And the mechanism, as I said, they probably, these are different amino acid substitutions, so they probably work somewhat differently. But with a couple of them, we have crystal structures, and we think they all prevent the movement of the DNA into the exonuclease site. One other thing that we observed about these mutants, that they're so hyperactive, so they can synthesize DNA at much lower DNTP concentrations than normal polymerases. So polymerases need DNTPs to make the polynucleotide chain, and they are present in cells usually in very limiting amounts. The cells have just enough to synthesize DNA. So this experiment was, we looked at DNA synthesis at the normal S-phase DNA concentration, which is 1. And then we can actually, in yeast, we can measure exactly the intercellular S-phase DNTP concentration. So we mimic these concentrations in the tube. And then we believe we see the most like physiologically relevant amount of synthesis and physiologically relevant error rate. So what we did here, we started with the normal S-phase concentration, and then we titrated that down to only 10% of the endogenous level. And the wild type polymerase gets inhibited very quickly as you reduce DNTPs. Then exonuclease deficient polymerase, which are not seen in cancers often. They are somewhat resistant because they don't excise anything. They don't need as many nucleotides. But the cancer variants, all these things in red here are cancer variants. They synthesize much better at the normal concentrations. This is what I already told you. But they are also very resistant to the reduction of DNTP pools. And there were a couple of variants, including the most common PR variant, that at 10% DNTP concentration synthesize better than the wild type polymerase does at the normal DNTP concentrations. So why this is so interesting? The cancers, presumably in early cancer development, go through a stage when DNTPs are depleted. Before the tumor starts growing really well, they go through this DNTP depletion state. And maybe it is very important at this stage to have a polymerase that is still able to synthesize DNA when DNTP pools are low. And I think I have our overall thinking about what all these polymerase mutations do in tumors. They, of course, they reduce proofreading to various degrees. They also increase polymerase activity. Both these things contribute to the mutator effect. So this accelerates the accumulation of mutations in oncogenes and tumor suppressor genes and gives rise to a tumor ultimately. So this is like a standard way to think about it. Now we have a theoretical way of thinking about it. That now that we know that these polymerases also tolerate low DNTP concentrations, so maybe they help beginning tumor cells survive this stage when DNTPs are low. So more than just producing mutations, they also promote tumor growth under these metabolic stress conditions. This is an unconventional role, and I even have even a more theoretical thought that maybe the mutator phenotype is just a consequence, and this is the main function. So this is why we get hyperactive, well-fit polymerases in tumors. All right, so now I'm going to switch to the last topic in my presentation, and this is the topic that brought me to collaborate with Anna. We were interested in the genomic features of these tumors, and the reason we were interested, well, we are generally interested in genetics and what happens in tumors. But it is important to be able to identify these tumors for clinical purposes by their genomic features, to classify them as poly mutant tumors. So to start everyone on the same page, I didn't know how many people will have background in mutagenesis. So these are all the different types of mutations that can potentially happen. There are base substitutions. There are 6 types of them. All possible substitutions of one nucleotide with the other, and I only list the purine base. The opposite strand will contain the pyrimidine. So there are a total of 6 types of base substitutions. Then there are so-called indels, which stands for insertion plus deletions. So either insertions or deletions, and these are usually small additions or small deletions, -1, +1, and so on. And then there are larger arrangements: deletions, insertions, inversions, duplications, amplifications, translocations, different kinds of complex arrangements where many different mutation types are combined. And finally, a chromathripsis, where the whole chromosome gets shattered and then joined together in a random manner. So these are all very interesting mutations, but POLE mutant tumors don't accumulate any of them. The only two types that occur frequently are G to A and G to T based substitutions, and G to Ts are most like indicative of a POLE signature, so to say, because G to A are very common type is a very common type of mutations. Many tumors accumulate them. G to T are more specific to POLE tumors. And then, other than the type of base substitutions, we also have to consider where, at which Gs these mutations occur, and not all Gs mutate equally. And one way to look at this, which is a traditional way in the field, is to look at so-called trinucleotide sequence context. This is when you consider the mutating gene, in this case, or if there's a different type of mutation, a different mutating nucleotide, and then one nucleotide before the mutation and one nucleotide after the mutation. So there are 6 different types of base substitutions, and if you consider all combinations of 5' and 3' nucleotide, you get 96 different trinucleotide motifs. And by quantifying mutations in each of these motifs, you get this signature 10, which is considered to be associated with POLE mutant status. You can see only 2 high bars here, and these are G to T mutations in AGA context and G to A mutations in CGA context. This is basically all that these tumors accumulate. The problems with the trinucleotide signature, it is a purely mathematical approach to analyzing signatures. Mechanistically, there is not a single process that I know that would specifically look at one nucleotide before the mutation and one nucleotide after. For example, DNA polymerases look at least 10 nucleotides before the mutation. They contact 10 nucleotides of the duplex DNA upstream of the primer terminus. So this approach, it is widely used, and it has been helpful for exploring some of the mutagenesis pathways, but we wanted to take it differently. And several years ago, we looked at just 25 random G to T mutations in a cancer cell line that contained a POLE mutation, the same P286R, the most common variant. And we saw that all of them, with no exceptions, were mutations in runs of multiple purines, different length, different composition. So this was quite intriguing to us. So we wanted to take it seriously. And this is when I emailed Anna and asked whether we could collaborate to look at this at a larger scale. So Anna and her group came up with this approach where we designed, and I'm sure some of you have heard presentations of this project, but I will give my perspective of what was interesting and how it worked for us. So they came up with a set of 104 motifs that are basically runs of multiple purines, starting with shorter runs of just 2 purines and up to 14 purines in a row. And this is like the long runs were just not frequent enough in the genome. And then in each category, we had the mutation position change from the beginning of the run to the end of the run. And we evaluated the frequency of these mutations in almost 1,000 of tumors, endometrial tumors and colorectal tumors. These are the tumors that where Pol epsilon mutations are the most frequent. And what we observed was very fascinating. So this is a graph where these motifs are arranged from the shortest to the longest, and the mutating nucleotide migrates from the beginning of the run to the end of the run. So indeed, the longer runs mutated more than the shorter runs. So you have to have at least 6 purines to have a true hotspot. And the other intriguing thing was only certain unique positions with within these runs mutated, approximately about 3 purines from the end of the run. And also, when the run gets longer, you start getting a second peak about 4 purines from the other side of the run. So I think we discovered something, and this signature is very specific for poly mutant tumors. Tumors without poly mutations don't have these mutations in polypurine runs. So I think we discovered here something very interesting about the polypurine traps in the human genome. We don't quite understand what it is, but there is something special about these positions that mutate and how polymerases handle these runs. And it will probably take a long time to understand the biology behind it, but we also thought that we could use this signature to try to distinguish poly tumors from non-poly tumors and try to use this for clinical purposes. So Anna's group developed a classifier that used this signature to separate poly tumors and non-poly tumors. And then we applied the strain classifier to tumors that had poly variants of unknown significance. And if you look back here, the tumors with poly variants of unknown significance are here. And there is a sort of something that looks like this wave here. And we thought this could be because this group of variants of unknown significance actually contains true drivers. So the classifier flagged 3 tumors with poly mutations as carrying possible unknown drivers, and 2 of them had many mutations. I told you every gene is mutated in these tumors, but sometimes you can have 10 mutations in the same gene. So this is really crazy. But this is where yeast genetics comes really handy. We modeled each non-silent mutation individually in yeast, in these 2 tumors with multiple mutations, and measured the mutation rate to see which of them was the true driver. And surely enough, we found 1 driver mutation in each tumor that was flagged by the classifier. That was satisfying enough to see that the classifier was very efficient at identifying the driver mutations, but we actually discovered something even more interesting here. If you look again, I told you that all previously known driver mutations map to the exonuclease domain. One of our new drivers maps exactly in the same place, but the other map actually to the polymerase domain. And this is the first driver mutation that we see in the polymerase domain. So how does it affect the accuracy of the polymerase? Remember the balance between the polymerase and exonuclease activity. So what we observed about this mutation, and most of this will now be a hypothesis, but this is how my brain started thinking immediately what the mechanism could be. So one thing we noticed that the mutation is located at the protein DNA interface, and there is a region in the polymerase that is very important for interacting with the DNA minor groove, and interactions with the minor groove are important for polymerase fidelity, the accuracy of DNA synthesis. And the mutation is here, and it changes glutamate to a glycine. So it removes a negatively charged residue. So it increases the overall positive charge in the exonuclease domain, and DNA is negatively charged. So one hypothesis is that it increases it stabilizes DNA in the polymerase domain. So basically, it achieves the same thing that exonuclease domain mutations achieve by preventing DNA transfer to the exonuclease site. So it achieves the same thing by drawing DNA into the polymerase site. So this polymerase will not proofread even though the exonuclease domain is intact, because the polymerase is always in the polymerase site. It is a hypothesis. We also have others. So invite me again in 5 years. I'll tell you more about how it works. So I think that's where I will stop. And I just summarize the data that I showed you today. So we found and validated 16 now poly driver mutations by mutagenesis assays in yeast, and our lab actually maintains probably the most comprehensive list of driver mutations. If there are any clinicians here that could benefit from this information, I'll be happy to share it. And then mechanistically, we see that the pathogenic variants are hyperactive polymerases with the polymerase to exonuclease balance shifted toward synthesis. And we think it's not the exonuclease defect that determines the pathogenicity, but the polymerase fitness, the ability to synthesize efficiently, including at low DNTP levels. And then regarding genomic features, poly mutant tumors accumulate as G to T transversions in polypurine tracts, and we have a classifier based on this signature that can efficiently distinguish between poly tumors and non-poly tumors. And this led us to discover a novel class of mutation, driver mutations in the polymerase domain. And I think that there will be more mutations of this class found. We just need to know where to look. And finally, I want to thank all my lab members that generated the data that I showed you today here. And this was an exciting collaboration with Anna's group. And the true reason I came here, of course, it was a pleasure to meet everyone in person, but I wanted to learn a little more about Anna and see if there are any fun facts about her that I can, showcase in the following talks, like if she could build a canoe or something. And then Igor Rogozin at NCBI was also an important contributor to this story. So I circled this collaborative project, but also I'm very thankful to Eric for his crystallographic work and to my multiple clinical collaborators that provided information about the mutations in patients and expanded my understanding of what kind of polymerase mutations actually occur in tumors and in the germline of these patients. And some other people contributed valuable insights and reagents. And thank you again for inviting me.
Thank you, Polina, for this actually, I would say, elegant talk. Yes.
Thank you. Thank you.
Yeah, elegant scientific talk. So please, question. Right.
If you could go back to the second part of your talk where you're showing her your model.
The crystal structure. Yes.
One or two slides before this.
Yes.
One more.
No, there wasn't a.
There was a figure that you showed that said the cancer-associated E301R had a lower mutational frequency than the your like control mutation where you deleted exonuclease.
This?
Yeah, that figure. So with the in vitro fidelity, I was just curious because you said that the exonuclease activity was ablated, so it should behave the same way. Was there a mechanistic explanation why the mutational frequency was lower?
The cancer mutation has a little bit of exonuclease activity, so it can proofread some mispairs, not all of them. So when you completely lack exonuclease activity, you don't correct anything. So you will have more errors. So this is exactly what we see here. When you don't have exonuclease activity, you make more errors in vitro. This is during in vitro synthesis. In the PR mutant, there is a low frequency because it can proofread a little bit. Does this answer you?
So the model, I think, showed that like the DNA is not able to access the exonuclease.
Yeah, well, maybe there are other confirmations. So the crystal structure is a snapshot of one particular stage. The protein is dynamic. Maybe this arginine residue can move out of the way. That's the only thing I can imagine. Maybe Anna could have more insight into how this could happen. But I think so in this paper, the crystal structure paper. As part of this paper, also there's molecular dynamics of polymerase DNA interaction there. So this determines the probability of DNA being positioned in the exonuclease site. It is improbable, but not impossible. That's how I understand it. Yes, I see there is some discrepancy between what I said. It mostly cannot be accommodated to the exonuclease site, but that apparently at some rate it will.
While on this slide, the second most common variant in the SNP was B something R. Is it in the same sort of physical space as that? Or
Yeah, no, it is actually on the other crystal structure. I showed it for a different purpose. Here, this is this valine residue. So it is exactly on the path that the DNA would take when it moves from the polymerase to exonuclease site. This mutation has been puzzling us a lot because valine to leucine is a very small change, one carbon atom, but it makes a huge difference. So we think that valine to leucine mutation prevents this movement from the polymerase. This polymerase, actually, when you purify it, it has almost perfect exonuclease activity because the exonuclease domain is essentially intact. There is no physical block, but the block happens here. DNA would not move. So it has perfect exonuclease activity, but when synthesizing DNA, it would not use it. You can do experiments where you compare, you give the polymerase nucleotides and you give it an opportunity to proofread, and you see what it will choose. It can proofread, but it will not. So this is what we see with the VL variant.
So one thing I didn't quite get, when you're doing these structure function experiments and these, you're putting the human gene into a yeast itself?
No, we make the same mutation in the yeast gene. Okay. Yes, and we have to have the region absolutely conserved to make sure that we make the same mutation. And in most cases, we can do this.
Has it ever been tried the other way? Putting human genes?
Putting the whole gene? Not really, but the prediction is that it will not work because of the domains that are needed for species-specific interactions with other proteins. What we are doing right now, we are making a chimeric gene. So the polymerase has the exonuclease domain, polymerase domain, and then a huge C-terminal part that is necessary for protein-protein interactions. So we keep the C-terminal part from yeast, and we put the catalytic portion from human. And these strains survive. So this would be the next thing that we will do. Because then you can model all human mutation.
Yeah.
Great talk, Kathy. So when you meant exonuclease position, does it mean that the whole exonuclease is knocked out?
Yes, you mutate the catalytic residue, so it cannot excise any nucleotide.
Does it mean the DNA will still go inside of nuclease?
Yes, it will still go. So this polymerase remains slow because it still undergoes partitioning, and it is slower yet because when DNA is in exonuclease site, it can't do anything. So it just sits there waiting to be proofread, and nothing happens.
Given that the whole exonuclease is knocked out and the whole unit is missing?
Well, in this particular mutant, the catalytic residues are carboxylates, aspartate, and glutamate, and they are mutated to alanines, just to remove the side chain in this mutant. But there are some others that we also, you can mutate aspartate to valine or to asparagine and get the same thing. No catalysis at all.
Right, this is a very different question, but I think this is really interesting from the perspective of what you said earlier, that people with these mutations actually have really big prognosis. So is there any interest in making polymerase faster as a way of treating cancer? Especially because you said they're also very insensitive to those synthesized on.
Yeah, I'm actually asked this question a lot. How we can it would be ideal if every tumor was a polymutant tumor, then they would be easily cured. I don't know how to do this, but yes, people are thinking about it.
Maybe question for Maria. So in general, if these tumors are hypermutable and they will be recognized more efficiently by the immune system, right? In
Yes.
general, but then these mutations should be beneficial for tumors as well. So is it because of the stage of their cancer development when these mutations are more?
Yeah, so I think, well, as far as I understand immunology, you get T lymphocyte infiltration later in tumor development. You don't get it when the tumor, they're at first beneficial. So the tumor doesn't start growing until you have mutations in oncogenes and tumor suppressor genes. This happens early. So when you are still not a cancer, and only you get a poly mutation, then you accumulate tumor suppressor and oncogene mutations, and then you start growing. And then you get T cell infiltration. And then you get the immune response, right? So they're temporarily different stages.
Are there any other questions? So are there any, well, I guess I sort of know and don't know. In terms of co-occurring mutations, we know that P53 was poly, yeah? What do we know about this co-occurring mutation between poly and other genes?
So these are colorectal and endometrial tumors, and particularly colorectal cancer progression is very well described. You have to have APC mutations, KRAS, P53, and they accumulate all the same mutations, but sometimes the types of mutations would be different. Like normal non-poly colorectal cancer will mostly have APC truncations from frameshift mutations in those. And then poly mutant tumors will mostly have missense mutations, but they have to mutate the same genes. So the spectrum will be different.
Just gonna follow up to that. What's the time that you're talking about? So is it possible to give a sort of diagnostic to prevent this type of time and scale is such that you know you have the mutation there, then you're gonna have you're gonna have mutation.
If you have a germline mutation, right, then yes, definitely. As soon as we know that the patient has a germline driver mutation, they will be monitored regularly, and well, they will. I mean, the tumor will be excised before it's too late, right?
There are programs for neonatal. I mean, there are programs.
Yes, but the majority of these tumors are sporadic tumors, and people don't come into the clinic until they have.
I'm thinking about the future.
The future.
No, I mean we have like in the
I've been thinking about that. So what would you do? Would you biopsy healthy people to see if they have a poly mutation in their tissue?
For newborn babies, there are studies that
Newborn babies will not have these mutations. It takes a while.
Yes, that's my question. If it's a
What
germline
age?
mutation, when do you think it would be good to look for this and diagnose it? Like for instance, we do colorectal cancer checks.
The germline mutations you can do at birth, because they always have.
So that was my first question.
Yes, but this is the minority. This germline cancer predisposition syndrome is not that common. And usually it is diagnosed when a person comes into the clinic having colorectal cancer at 30, for example. Then people start thinking, so something is wrong. They start sequencing the genome. Nobody sequences newborns because the rate will be very low for these poly mutations. For people with most poly-mutant cancers are sporadic cancers. So there is a time period between like getting a poly mutation and developing a tumor, but then it is only in one cell. How are you going to find it? There's no straightforward way. But I think at this point, the most clinically significant thing is to classify these tumors as polymutant and treat them differently. Because they actually, these are the tumors that take care of themselves.
Time for one more question, Peter, please.
So you were asking at one point, you know, if there are people that do clinical work and interested in certain aspects of it, and got me to thinking. So some kinds of mutations, like microsatellite instability, there's a subset of genes,
Yeah.
that are likely to be mutated in those. But of course, there's lots of different players in DNA replication as well as repair. Is it at a point right now where you can look at the mutation frequency, or not the frequency, looking at the type of mutations, predict what kind of defects there are, which genes involved in repair or DNA replication are the ones that are mutated or dysfunctional? Because I think of this in the context of these variants of unknown significance, because in some cases they probably aren't really causing a defect in functionality. So can you match up the mutation profile with
the causative mutation? To some extent, yes, definitely. If you see microsatellite instability, this is a mismatch. This is well implemented already. Now we know that if we have a high rate of mutations in polypurine tracts, this is a poly mutation. This is a very characteristic of poly tumors.
Is that the case with other genes?
I'm thinking if there are any other examples. There are signatures of exposures for sure, like UV exposure or benzopyrene exposure or exposure to the most common mutation now known appears to be aristolochic acid, which induces a very specific type of mutations, AT to TA, that are generally very rare. So this can be, the exposure to this substance can definitely be. There are a couple of others, but they're not like DNA repair defects that we are talking about. Because the repair systems prevent so many different types of, they repair so many different types of lesions. So I don't think there's a unique signature. I would say that G to T transversions are also indicative of oxidative stress or defect in oxidative damage repair. But this is more difficult to connect.
So Polina will have 15-20 minutes, will be still here. If you want to talk to the please, here. And we would like to thank the speaker for coming. Thank you.